So welcome everyone. It's a real pleasure to be here today and to see such a crowd full of people. It's absolutely amazing to see the room full of all of you. And I know it's early morning and I think I've got a lot to say, a lot of exciting things to tell you about. Probably more information than you can digest in the hour, but I do hope that you will ask questions. If you have still got more questions afterwards, I'm here all day. Please don't hesitate to find me, and I'll try my best to answer any questions you may have. So I'm going to launch straight into the presentation today, and just to tell you that there is such excitement around the research, and I hope in this time that I have, I can convey that. Let me just... Certainly, things have changed significantly since the Ice Bucket Challenge, and everyone here has their own story to tell about it. I hope today I will share some of the sentiments that I had around it and the impact that it has had for research. And things that come to mind, of course, for all of us, is a huge hope, a hope that now there is much more awareness globally of the disease. It certainly has come with increased funding globally, which is incredibly exciting. It's definitely come with increased research interest. I have just in the last 3 months had over 600 different ideas and proposals that are in review for studies, just in 2-3 months. Huge opportunities, and that's what I hope I will convey to you today. Certainly, there's a sense of urgency and an anticipation that something is coming soon, and I do believe we have some fantastic things in the pipeline. I also believe there's a lot of frustration, because all of you want those answers right now, and I'm here to tell you that I think there are amazing things. We are going to make a huge difference. We are moving forward. Sometimes it doesn't feel like it in the time frame that all of you want the answers. And so I will give you a sense a little bit why there are challenges to find treatments and a cure for ALS. But that doesn't mean to say that we can't get there. So what I'm going to do today is speak very briefly about ALS. And of course, everyone living with the disease, families connected with the disease, you know more about the disease in many aspects than I do. But I think to try and understand how we as scientists move the field forward, it's helpful to think a little bit about what the disease looks like from a biology standpoint. I will then talk to you about the opportunities, the huge opportunities, but also the real challenges we have to develop therapies. It's realistic. It's no point to tell you now it's going to be an easy job just because we've got some extra funds. This is the reality for drug discovery. I'll talk about why, because of those opportunities and challenges, we've built our research strategy the way we have. And I speak a lot about we, I might speak about I. In reality, it's a combined effort. I have global advisors, I have teams of people who think about how we might do this. There is no one right way to do this, to spend money. We all know that when we invest our money in a portfolio. But there are ways that we can do it as best we can do it in the most productive and strategically sensible way. And I will impart what I think are my ideas and my colleagues and advisors who have helped me do that with me. So I think many of you have seen this slide. I like to use it. It gives you a sense, and I've just reminded myself that I'm not going to do much pointing because I think I might fall off the stage at the angle that these things are, so that would be interesting. So I'm going to try and describe. But what you see here in front of you is really the challenge of ALS in terms of its biology. It's not one focal site. ALS is an upper and a lower motor neuron disease. These nerve cells connect exquisitely with the muscles that they are supposed to contract and interact with. When things work well, they work exquisitely well. But when things go wrong, and that can be in multiple places, in the upper motor neurons, in the lower motor neurons, in those long connections, and then in contracting those muscles, the disease occurs. And so how do we fix that? It's not an easy fix to just replace something in one specific spot or place. Nerve cells are remarkable cells. They are the longest cells, the motor neurons in the body. They have a small cell body where all the information and messaging is. They have these long projections that project to the target, in this case the muscle, and all the things in between to make sure that the messages are occurring accurately and correctly. So a lot can go wrong and does go wrong in many different parts of that system. What we understood very long ago was that the motor neuron, that nerve cell, is central to the problem. And if you see these two images here, you have a normal motor neuron cell body that is looking healthy. Below it, a rather shriveled version. And these strange inclusions and abnormalities that are happening in the neuron, we've known for years that that's what it looks like in the person's tissue. But now, what does that mean in terms of the disease progression and what is happening? And we always focused on that motor neuron, but in reality over the years, we've changed our view that it's not only that system involved, but if you look below at the red cells, those are the astrocytes, very important support cells that sit around those nerve cells. They regulate things like glutamate, a very important transmitter in the brain, but if in excess can cause problems. And then if we look to that image there, that what I call the Christmas tree image, a beautiful nerve ending, so that each motor neuron, each nerve, exquisitely hits one point on that muscle, but actually with a very complex network as well. So if any of that shrinks and shrivels, those connections are lost. So the complexity is beyond just that motor system, but the cells that surrounds it and the cells that they interact with, and in particular the muscles that they are contracting. So then our problem gets even bigger, because we tell you when we talk about motor neuron being a nerve system, muscle system disorder, but actually in conjunction with it, there are systems that are involved that cause a problem for the cognition part of things called frontal temporal lobe dementia. And so this component is a real component in some cases of ALS. So we have a spectrum. We might have a pure ALS, which is purely motor systems. We might have a pure frontal temporal lobe disease, which is not Alzheimer's disease. It's actually quite exquisitely different to that. Cognitive changes, behavioral changes that are very specifically defined, different from Alzheimer's disease, and then you get a combination of the two. And this really was crystallized by the discovery of the C9 gene, and I'm going to move to that discussion later. So we know that this is not just a potential phenomenon, we know that these combinations happen. And why is that important? Well, one, it's important because of the biology. We have two very separate systems involved. It's also because it's going to impact how we think about the care for someone who has both frontal temporal dementia and ALS. And it might impact the treatment paradigm, whether some treatments actually won't be as effective in the combination or will be, and how do we design those trials. And then we get a picture again that you've seen many times, and people have many versions, and I can tell you when I go to a conference on Alzheimer's disease or Huntington's disease, this picture is strikingly similar. The only thing we change is the cell that's affected. So in this case, the motor neuron is central to what is going on in disease. In other cases, in Alzheimer's disease, it's a different nerve cell that is involved that is particularly interesting and central to it. And so what is happening is that there are multiple things going wrong. You have your support systems there, the astrocytes. You have the microglia, which are the inflammatory cells of the brain. We have issues with things going from that cell body, that very long distance down to our spinal cord, so the transport system, and there's an important scaffolding that is required for proper transport of things. We know that things are going wrong at the muscle end. Is that early? Is that late? So there's a whole host of things going wrong. I think this is the most important key message to you when we think of the challenges of treatment development. What part of it, how many parts of it, are we going to be trying to tackle to treat? When are these things going wrong? What are the earliest things going wrong? By the time you go in to see your clinician and you've gone through the time frame it's taken to now eliminate other things that might be causing what your symptoms look like and say you truly have ALS, what has already happened? What are the important things that we should be thinking about treatment? And how can we reverse the disease? So in that complicated picture are all our messages of our challenges, is the focus of our portfolio, because we have to figure out every single one of those problems and see to what extent they're important and at what time they are happening. And it is our opportunity, because those are the handles in to thinking about how to start treating something. So why do drugs fail? And everyone feels that this is an ALS problem because it's most important to you here sitting today. Actually, it's a huge challenge for Huntington's disease, for Alzheimer's disease, for Parkinson's disease. it is challenging to find a treatment that is really effective. Why? I'm going to talk about the word target quite a bit. A target is simply to say, you need to know how to design your drug. Your drug has got to attack something that is going to change. So your target could be a specific gene mutation, which we'll talk about. It could be a system called, for example, inflammation is your target. Inflammation in itself is a whole lot of pathways, but those are targets to try and hone in on. We might, when we do a clinical trial, be testing the wrong target. It might not be relevant, especially not relevant at the stage and time of the disease that it's occurring. It just might be the wrong drug, or it might be the right drug that's just not getting there. So we're not getting to the target. So the drug, the design of the drug, the fact that many drugs don't go through the blood-brain barrier, because we do have exquisitely a protective barrier for good reasons, but then if we can't actually get into the brain, it could be a problem. So we think more carefully about how do we deliver that treatment. A big, big problem for us is actually have some of the drugs failed purely because we couldn't really measure their effect. So we use a score, which some of you are very familiar with, the ALS FRS score. It has been a good score for us. It's one way to measure whether things are changing over time. But it's not a measure that we can put a quantity on. It's more a quality of how things are getting worse for a person and if a drug can slightly change that effect. So it's not a hard and fast measurement. And I repeated this already a few times. Are we starting to try and treat at a point where too much damage in that particular pathway has been done, so it's very hard to reverse? Maybe we'll slow it down a bit, but we can't really impact it as effectively as if we knew much earlier that the disease was happening. And most importantly, and I think all of you can attest to this, not everyone has exactly the same disease. At least it appears in very different forms in different ways, some slow progressor, some fast progressor. Is it more like in the cancer field, where we divide cancers in very specific types of cancer? And are we putting ALS into one bucket, but actually we know that there are very different forms of ALS? And this is not a new phenomenon. The clinicians are thinking about this now over several years, but it might well explain why some trials are failing because we're binning everyone together. It's not an easy problem to solve, though, because how are we going to divide you as a population? Is it because of the gene that you carry? Is it because of the exposure or environment? Is it because you're slow or you're fast? So these are not easy answers, but there are real problems for why drugs fail. And also importantly, do we think that one treatment approach is going to be enough? Is it going to be a complexity? And I think I'm sort of answering that question. It is complex, and therefore it's likely we need multiple treatments. And it is the way we're going to go. We have riluzole. We're not excited that it doesn't have a huge impact, but maybe riluzole plus and plus and plus will eventually, in combination, like an AIDS cocktail, have a much more meaningful effect. And then we look at drug discovery in general. This is a version of many versions you would see in the industry and elsewhere. Drug discovery is a fairly systematic process. You have to have A to get to B, to get to C, to get to D. Having said that, even the industry, recognizing that things are not being as successful as they want to be and need to be, are thinking of much more creative ways to try and change this. How can we at the association, with what is a fantastic influx of money, but certainly not enough. If you see on that slide, it takes $2 billion to get one drug to market, and we don't know if that is going to be a successful investment to get it to market. So how can we actually impact this with significant funds, but not all the way there? We haven't got all the funds we need. Well, first of all, I will say we will leverage funds, because small things can bring big funds towards those successes. But the other important thing is we can probably do more to define the target, the way that we're going to attack this. And that we can do by improving our biomarker development. I'm going to spend time on biomarkers. Gene discovery is pivotal, and I'll tell you examples of 2 genes that are definitely going to be much more successful in driving therapies for those 2 genes and maybe others. And then disease modeling is important, because disease modeling, and I'll go into that a bit more, is important not only to test ideas downstream, but also to further support our initial hypothesis of how the disease is working. Then we can do much more, I think, in the terms of our preclinical modeling. We'll talk a little bit more about what the limitations are of models and how by combining multiple models, we are much more likely to have a successful outcome. And then importantly, I think the big challenge is that so many trials that have a smattering of some potentially interesting thing at phase 2. Really, we haven't had any blockbuster effects at phase 2, don't be misled. We have had some smattering effects. Are those studies well enough designed to be sure that the effect is real? And our limitation there has been the lack of biomarkers, the lack of careful trial design, not through a community that isn't trying to do this and a community that has done many trials and has learned. The clinical community is very committed to doing it right. But I think in combination with working with researchers, individuals who are thinking about these disease-modifying approaches in very early development, together with the clinicians, we need to develop much better clinical trial designs. And I don't have time to talk about it, but we're very excited that the organization is very involved in this, and you'll hear more about it, is developing an FDA guidance document. And I will tell you that the clinical and research arena is extremely excited and very behind helping us build the right information for this guidance document. So what are our strategies? And I won't say to anyone now that we reinvented the wheel after the Ice Bucket Challenge, because the research is the research. The ideas are important ideas. So we have based it on what was already in existence. We had established over the years a translational program. Very important to do good science in the lab, but absolutely useless if you can't get it out of the lab into the clinic. So how can you do that, and how can you push that? And the nonprofit world, like organizations like the ALS Association, have a huge role and ability to play, because we can really drive the right partnerships. It's all about pushing academic and industry partners together. Academics are brilliant at ideas. I've been in that space. You think about things, you develop things, you keep on doing more studies to make it better, but often don't have the drive and direction of getting a product at the end. The industry, we might hate them or love them, but the reality is they know how to do the business of getting a drug to the end space. They haven't done it for ALS yet, except for riluzole, but they've done it for many other disease areas, and they can do it. So bring those two together, and I think that marriage brings a much better opportunity for success. We have also, over the years, and our centers of excellence are a pivotal part of this. You people living with the disease are a pivotal part of this. We have that in our organization, in our partnerships, in our collaborations, and that is what is going to drive treatments forward. And then I believe it is all about partnerships. It doesn't matter if the discovery is going to be made in Ireland or in Portugal or in Australia. We want to be part of that team and help drive those discoveries forward. So we've always had a very global program, often take on board very risky, crazy ideas and help them either move or kill them if they're not going to work, and really believe in building more and more consortia, we will get to the end goal. So just for ease of describing what we do, we have really 5 strategic areas, and I'm going to touch on each of them very briefly. So just to re-emphasize, we haven't reinvented a wheel. I'm not going to tell you that science is different now since the ice bucket, but we have funds that can move things in a much more meaningful way forward. We have been able to make partnerships that we could not have done before because we didn't have the funds to do it, and we can really drive some of our initial programs that have made huge discoveries in treatment approaches and do many, many more of them at one time. So gene discovery. This is what the arena looked like when I joined the field in 1993, the first gene to be identified, SOD1. It looked rather bleak for many years. It was very hard to find the next genes. But then amazing things happened. Over the years from 2004 onwards, we have a plethora of genes. Part of that is technology. Part of that is a discovery that actually some of the modern technologies don't always work, and going back to the drawing board with some of the older systems helped us find the one very prevalent gene mutation, C9. And so what we know for familial ALS, and I don't know if my colleague Dr. Traynor is in the room, but he has been one of the key players in driving this forward, both by his science, his clinical work, and the work he does to collect samples and find the genetics of ALS, both in sporadic and familial. But we now have over 60% of the genes that are found in familial ALS. ALS for which there are probably more than 2 people in the family that carry ALS. It goes most of the time from one generation to the next, although there are situations where it is not highly penetrant. So familial is when it is in the family, as opposed to sporadic, when the disease is not really clearly linked to one single gene. I will say that we quote familial ALS to be about 5 to 10%. You will see different numbers because in reality that 5 to 10% is not entirely accurate. It's a generalization, and in fact, in some areas in the world, the percentage of genetics is much higher. So it's very, very variable. In some parts of the world, you won't find a particular gene. In the Netherlands, there are no SOD1 mutations, for example. In Italy, there's a high percentage of C9 mutations. So partly how populations have moved and how things have changed over time. But so it's important to note that the genetics plays an incredibly large role in all of ALS. Because of that, our major first initiatives were to fund genetics, because we felt very importantly we need to find all the other genes. But with genes comes an opportunity to collect samples from patients, to get much more clinical information, and to link now much more effectively clinical details with the genetic details. And oh, by the way, we can also collect from each single person skin cells so that if there is some interesting genetics in that particular family, we can develop model systems from that family in a dish, and we'll talk more about it. So the New York Genome Center is a hub, is a centralized space that has the latest technology, but it's not isolated to New York. It's built around there to open up researchers worldwide to use that facility, to partner with us, and all the data is immediately available to any researcher who wants to explore. So it's very open source, it's very collaborative, and it's meant as a place for genetics for worldwide collaborators. Project Mine is another initiative that is driven, actually very interestingly, by a person living with ALS who felt compelled that genetics needed to be found out for not only familial but sporadic people, and that every single person should really have their genetics sequenced. And because of the cost of sequencing, that's become much more of a reality. The importance of Project Mine is that it's a global initiative. Again, as I emphasized, where in the Netherlands there are no SOD mutations, there might be a unique genetic there that we're not picking up by doing an only US-centric focused study. So by bringing genetics as a global study, we will find very interesting genes linked to the disease. And our colleagues John Landers and Jonathan Glass are the US partners to the global project. The goal of the project is to sequence over 15,000 patients and controls using a technique called genome-wide association studies, and that is where we combine lots of different studies together. But more uniquely now, and a technique I don't have time to go into detail, we can now look at the whole genome, which means it's the genetics that codes for all our proteins in the body, and a huge part that is actually doesn't code for anything, and we kind of threw it away as being unimportant. But actually, there are clues in that part that doesn't necessarily encode for proteins that is very important. And we learned that when we actually threw away the stuff that we didn't think was important in the C9 mutation, because it came in a region that doesn't encode a protein, and I'll show you that. So this is newer technology. It could not be done before at the same scale because of the cost. The cost of sequencing has become such that it's realistic in a short 2 to 3 year time frame to sequence 15,000 ALS patients and their controls, because that's very important. You have to have very good controls because we all have variations in our genetics. What makes one person have ALS versus another person with the changes not have ALS. So it's got to be a gene specific to the disease, and that takes an enormous amount of work because of the numerous changes we just have unassociated with the disease. So there are many, many other genetic studies. We have several that are going to be announced in the upcoming months. These two I quote to you because they've been formally announced, but be assured that genetics is a key area. We are adding on to studies that are already ongoing in multiple parts of the world, and so there'll be a lot of announcements around gene studies, collecting important big data which will enable us to put lots of the pieces of the puzzle together and figure out the genetics of ALS. Importantly as well is model development. And I know that many of you have been involved and hear about the discussion and the frustration. Well, something worked in the mouse and it's failed in the clinic. I have two versions of that. I did build one of the mouse models for ALS, so was very involved in mouse model development. Everybody recognizes a mouse is not a human. I would say that the mouse models have been incredibly important in helping us understand disease pathways. I showed you earlier that we were very motor-centric. We thought the motor neuron was the most important. It's the mouse studies that really have told us how important the glial cells are and the inflammatory cells. But a mouse does not do everything. It's still a distance away from the human. So rather than saying the mouse is useless as a tool, let's go to all the other tools, we have no proof yet that the other tools are going to be a proof that this is going to work in the clinic. But use them in combination, and I think we have a very, very strong position to say if these things work together, if the pathway we are testing in that mouse or that fruit fly or that worm is of importance in the human disease, then it might make sense to go on. So what I'm trying to emphasize, although some will have out there in the field to say everything is now iPS cells and I explain them, I think they're a fantastic tool. But they may not be the only answer. So let's not throw away things that we have for things that we don't know for sure yet. Let's do them all in combination. Interestingly, I have the dog model there because this is an amazing finding and work that's being done by one of our colleagues. There are certain strains of dogs that have a spontaneous mutation. And lo and behold, that mutation is an SOD1 mutation. So we do have ALS in dogs. And so these dogs are being used to help us develop, because they are that step further closer to us as humans, especially also in size, because if we're trying to do gene therapy approaches or antibody approaches or antisense studies, which I'll all show you, the dog will be a better model. And so they are being used to help us, but also to help the dog owners to find treatments for those dogs. But most importantly, none of these model systems make any sense if we can't connect them to the human. So by improving how we can, in lifetime, see what is going on in a person living with the disease, these are images, for example, in the brain, we can try and correlate pathways with what we are discovering in our model systems. So we have many initiatives to develop better models, more models, strategies of how to work together with these studies and make more sense of them. This is a work by Dr. Finkbeiner. It shows you here how he's taking a skin biopsy from a person living with the disease. And then remarkably, you can take these skin cells, do some manipulations, and in a dish, drive them to become nerve cells in the body. You can get the astrocytes, you can get the muscles. So this is really now changing the era of stem cell approaches for many reasons. We can now be much more engaged with people living with the disease, because that's what it's all about, really. We want to make model systems not from mice and from worms and from flies. We want to ideally make them from the people that live with the disease. So as we get these systems working better, how reflective then in a dish is this of the human disease? And Dr. Finkbeiner is part of a huge initiative, the Neurocollaborative, and I know some of you might have listened to his webinar, has developed over time, and in fact, he was working in Huntington's disease much more prominently than ALS and became very interested to work with this in ALS, developed a microscope that can, in very short time, look at lots of these images and see over time how these cells in a dish are changing. If you then throw on it a drug that might be useful in the body, you can then see if that is improving the system. And then through very complex modalities, and taking from the left, you see all those raw images, enormous amount of data. How can you then make sense of it? And he is working with Google, and there are others working with IBM and other processors to really bring all that information through machine learning and other complex algorithm systems to make sense of it, to try and tell us what that reflects as to what's actually going on in a person with the disease. And this will more accurately help us determine whether a treatment approach is effective. Through the Neurocollaborative, through his partnership, we are going to build partners with companies who are then excited about the data coming out and the potential of developing that therapeutic approach. In partnership, as the Neurocollaborative is 3 laboratories in California working together with their outside partners. Clive Svensson has set up and developed a stem cell core. We felt it very important to have one centralized place where many investigators can bring their ideas of how to standardize the development of these cells. Excuse me, one moment. How to really bring the cells from one lab to another. It's just like doing a clinical trial. You have to work with clinical centers to make sure that your measurements in the trial are standardized. If we are starting to do studies where we're using iPS lines, but lots of different ones, it's going to be very hard to compare one from another. The team as a core are not doing it alone. They have all the experts in the field of iPS technology who are helping them to set the right protocols, to establish the systems that are going to be most favorable for people to share. So again, this is open source and open data sharing. He is also very involved, and that's why I highlight here on the right, in developing transplant studies. So I talk about stem cells and iPS cells as a huge opportunity and a tool for drug discovery. They certainly are being approached as well, of course, to use them as a transplant method. A lot of the work that is done at the moment with neural stem and others is really using cell systems that were developed in the past, but we are now moving towards trying to expand these skin cells and see if they too can be used as an opportunity for transplants. We're then going to move along to our program in drug development, and this is really a program that we seeded several years ago and had great success stories, but didn't have the funds to invest in significantly. And I'm very pleased to say that we can now really invest in a big way into this arena, because I think it's critical. And what I show here on the left is the funnel effect. And I think this is the value of the Ice Bucket funds, and this is a big part of our strategy. We have to do many things at the same time to hope that a few of these things are going to take us to the end goal. And so in this drug development contract program, for which in fact we got 90 ideas and selected 40 in for full proposal, we hope to fund around 10 to 12 of those ideas. These are all partnerships with the industry, so there are many biotechs and companies that are thinking about ALS, but they feel it's too risky to completely invest in it. So by giving them these funds, by finding them an academic partner to do the initial studies to make sure that there is a potential for this to be a drug that needs further development, we are basically de-risking and incentivizing the industry to be a partner. So we will first of all give small contracts to many, many studies and ideas to fill gaps where we need to. And then those few that look really promising, we'll take them all the way into the first phase clinical trial. And why I highlight these 3 companies, Biogen, Genentech, and ALS Pharmaceuticals, is because we've done exactly that with these 3 companies, and very successfully. So most importantly, I think, is our impact that we made on Antisense development. This was a small grant to Don Cleveland, Richard Smith, and Tim Miller at that time. No more than about $400,000, which at that time for us was quite big. It's all relative to how many funds you have. And this was actually Richard Smith's idea, a clinician who'd been very actively involved with caring for patients and had joined Don's lab for a period of time and said, gosh, if we were able to directly attack a gene mutation and get rid of it, could we have an impact? And we already knew that the company ALS Pharmaceuticals could do that for very specific diseases, but had never done that for a neurodegenerative disease. The one hurdle is getting this antisense, and I'll show you schematically in the next slide what that really means. But the hurdle is getting that system into the body and getting it to where it needs to be, the nerve cells, and shutting down the wrong gene in the right cells. And so he was able to, in a model system, in a rat and a mouse, put it into the ventricles, and I'll show you an image, and in fact it did work. And because of that finding, this technology is now used for Alzheimer's disease, for Huntington's disease. It's in clinical trial for a childhood motor neuron disease called SMA. And what it basically is if you have your gene, a gene is described into a messenger RNA. If you have a mutation in a gene, like SOD1, it then is going to transcribe to an inaccurate message and an abnormal protein. However, if you can then do something to block that message that is inaccurate. So you take this string of DNA and you make it align with the inaccuracy in that particular RNA molecule. You then can't get that protein made. It blocks the protein from being made. And therefore, if you don't have that abnormal protein, the hope is that you take away the disease. An important part of this, though, is to remember the mutation in SOD1, you get one from your mother and one from your father. You only carry one mutation, whether it came from which parent, it doesn't matter. So you only have one gene that is in error, and you have one gene that is correct. So in fact, if you remove half of the protein, which is what is going to happen in this kind of system, you still have healthy protein to do its normal function. This system only works on the basis that the protein that is carrying the mutation is doing something abnormal. I spent 4 years in Don's lab trying to figure out what that protein was doing abnormally, and I have many theories, and we all have our theories. But actually, maybe we don't care exactly what it does wrong. It's doing something wrong, so let's shut it down. And this is a technique which can do it, which has indeed gone all the way to phase 1 clinical trials, was safe in people. The only reason for SOD1 why it went back to develop new agents is that in that 10 years of discovery of this technology for brain diseases, we've made much better advances and we can do it much more effectively. So there's a new version about to go into clinical trial. And it's based on the fact that what we have in our brains, the ventricle, which we see on the left, that space, and in our spinal cord, there are spaces around the brain and spinal cord through which fluid flows called cerebral spinal fluid. And it's these spaces that enables us to take something like an antisense moiety all the way through to the right places. So that was a phenomenal discovery by Richard and then by others as to how these antisense systems would work for brain diseases like ALS. And then we discovered the C9 mutation, almost, in fact, more than 10 years after SOD1. And this was an amazing finding. It did multiple things. C9 is linked with frontal-temporal lobal dementia and ALS, so it really told us that there is a connection with ALS and this frontal-temporal lobal dementia. It affects 40% of familial cases, so almost half of familial ALS is C9. It can be more so in other countries. It affects at least 8 to 10% of people that have no family history. So it's a very, very prominent gene. It was discovered in 2011. And actually, having built this technology of antisense technology for SOD1, we cut out a lot of time and length of time that goes into discovery. And it is possible, it's not guaranteed, I can say it, but the company's not saying it, that in 2016 we would have a clinical trial. So within 5 years, we have a trial which sometimes takes 7 to 10 years to get to that same point. So this is phenomenal. This wouldn't have happened if we hadn't invested in that early study of 400,000 to make it work for SOD1. So I really relay this a little carefully to give a message to you. I know you want to all hear from me that between the ice bucket of August and today, we have made an amazing discovery, and tomorrow I have a treatment for you. I can't tell you that. It's not the truth. But I can tell you that our investment has an impact and will speed up things in just the same way that this SOD1 investment made a difference not only for ALS, but for at least 5 other diseases. So rest assured that with the clinical community and the academic community and the industry engaged, we can make a difference, and we will make a difference, and we will try to speed it up as much as possibly could be done. So I have to tell you about C9 because I told you that actually novel technology did not find this particular mutation. And that's what we must remember too. We often hurtle ahead to want to be sure that these sexy new techniques are it, and we invest heavily in it. We must do. But don't forget that sometimes it's simpler methods that need to be taken. And we spent millions on finding the C9 gene, and we spent many, many years and many frustrating teams of people because we could not find it. And it was because we were looking in that region, if you see in front of you the slide, from ATG onwards. We had a system that did beautifully at chopping out everything that we thought wasn't important, but we could look very quickly and at high frequency at things from ATG onwards. I'm being a little simplistic in my description, but for the terms of understanding the logic. And so we were missing that big fat orange triangle. And so 2 teams that we funded, Rosa Rademakers and Dr. Brian Traynor, again, I don't know if he's with us now, but he's coming later. They thought something is odd, something is awry, and he can tell it better than I can, and so can Rosa. But essentially, they went back to old technology and found this enormous expansion. That orange thing should be there in small size, but in people living with the disease, it's repeated over and over and over again, thousands of times. And just showing you a little bit schematically, normally, that's what it would look like. When it's expanded, you see an image like that. A remarkable finding, because it did 2 things for us. It found a gene that is going to impact a large number of people with ALS. It told us that often we think we're looking at under that lamppost where the things are supposed to be happening, and we're forgetting about the things on the outside. And so there are new technologies to look much more carefully at these kind of things that could be happening in other genes. So shifting away, just to tell you that C9, of course, we almost have monthly a publication on it. There's things going on all the time. It's going to be developed very fast. We are developing biomarker profiles. So C9, I could spend a whole hour talking about. But I'm shifting away from that. Happy to answer any questions. I haven't seen Mark waving at me yet, so I think I'm fairly good on time. So we'll keep going. So delivery was an important issue, as we described. And so although Antisense looks very promising, the same team as the part of the Neuro Collaborative with Brian Casper, who has done this beautifully for SMA, we are also thinking about gene therapy approaches. We have been for quite some time. This is work from my colleague, Dr. Zushang Zhu, and it illustrates to you one important point. And I know it's a complicated but beautiful picture, but what you need to note, first of all, gene therapy uses viruses to deliver something. The viruses are no longer infective, so you don't get an infection from the virus, but it travels to where you need it. And why have we not been doing so well in this area for ALS? Well, if you look to the left, it says, it is the left, AAV1, AAV2 are often used in clinical trials. If you go all the way down, finally, AAV9, so the 9th version of that vector, finally gets to where we want it. And if we look in that green area, this is just a mouse spinal cord. You see the big blob is the brain, and the long thing is the spinal cord. Finally, we have a vector that we can drive to get it where it needs to be. And so, shown again in a similar way, in the brain, we couldn't get, with the viral vector AAV1 through 8, sufficient of whatever it is you want to get to the neurons to the brain, and we can now with AAV9 and 10. And I will tell you that for spinal muscular atrophy, the childhood motor neuron disease, they have now already done this in babies and shown by delivering what they are missing in the disease, which is SMN, and that's another biology, they can deliver this to where it needs to be, and it appears, it's early days, it appears to be safe. So Brian Casper, who is really the leader in the SMA field with this, is working with the Neuro Collaborative to do this as well for ALS, together with Don Cleveland. So another partner to us, and also came through our program where we seed ideas, is a partnership with Genentech. And very complicated, but I will give you just the highlights. The goal here is to try and restore something occurring at the muscle. And that pinkness you're seeing and the square I've highlighted there, that's a connection point in with the muscle. That's a very important connection that seems to be lost or decreased in people with the disease. And Genentech happened to have on the shelf dormant therapy that wasn't being developed any further that could stimulate that particular connection. And so they thought they would try that with our funding and our partnership with Steve Burden to see if it works. And in fact, in our initial study, very promising, very exciting. The company touts it as a real example of an academic industry partnership. And the first phases have worked. We are now reinvesting in the second phase with the hope that once this study is complete, Genentech will take it over for further clinical development. I think I'm going quite slowly, and I'm going to have to move along a bit. There's a lot to tell you. So biomarker discovery. I've talked about it a lot, but it's so critical. It has many components. It's a signature of the disease, and it will help us improve diagnosis. It will help us to do what I said earlier, our challenge. How do we separate one population in a trial versus another? It will importantly tell us if we are attacking the target we want, a pharmacodynamic marker. It will tell us how the disease is progressing, and if when we treat the disease with something, that progression is changing. They can be images of the spinal cord. They can be physical images to the left, or they can be chemical readouts to the right. They can be brain images. These are all biomarkers. We can develop them with special marker systems on them. This is in a rat, and this is a marker for the glutamate transporter on astrocytes. Very exciting. We are now moving this into safety testing in humans, because if we can actually measure in people over time whether indeed there is a change in this particular luminescent marker, we can truly define and say, yes, glutamate transport is a problem. Not in every person with the disease, but in this patient population, and therefore drugs that affect glutamate transport will be effective in this population of people. And that is actually one of the considerations when the compound ceftriaxone that some of you might know about was tried. The trial failed. It looked fairly promising at phase 2, but we didn't have a good enough readout. And it might have been that some people might have benefited and others didn't. And we couldn't tell which person had a problem with that transport and others that didn't. And this will be able to do that readout. It can be a measure of strength. This is an Atlas chair that we have developed with Pat Anders and is being used actually at the Cedars-Sinai Clinic and several others to more effectively measure muscle strength. It can be a marker in the blood or the fluid. And I told you, one of the real problems is making sure we get messages from the cell body all the way to that muscle that it's innervating. And that is along a scaffolding that has to work exquisitely. Actually, when cells are dying, the scaffolding seems to be released. And this is called neurofilaments, parts of the scaffolding. And we now know that actually neurofilaments, there's an H and an L and an M, very complicated, but in sense, these scaffolds, they seem to increase in patient blood. And actually, it's an indication that cells are dying. So it could be a very good measure to see where things are in the disease. And in fact, if you look at many patients, these things can be very variable amongst patients, but they are increased. And interestingly, they stay fairly stable over time. But what's very important from the study done by Kevin Boylan is that if you have fairly high levels of neurofilaments in a small number of patients we've tested it, the progression can be very fast. And so can this be an indicator for us for people that have a slow disease progression and people that have a fast progression? Remember when we design trials, often people with a slow disease are excluded from trials. Why? Because it's a problem, because if we have a mix of slow and fast, we don't know if it's the drug that's helping that or if it's just the natural history of the disease. If we can define now our two populations of faster progressors and slower progressors, we can factor that into our trial design and not have that error affect our decision on that drug. Many more complicated technologies to show that there are differences in maybe immune profiles in blood. A very interesting, very recent study that we funded that shows perhaps that you can have a marker in urine that is effective. So much easier to get that biomarker source than others. And very complicated picture, but basically all you need to highlight there is, and this is where a pointer could be very helpful. No, wrong direction. There is a fragment that is removed from this particular receptor. That fragment, when released, can be picked up in the urine. It's an indicator that cells are dying, basically as simple as that. And it's early days, so it's not a promise that it's a proven one yet, but very interestingly, done by a group in Australia together with Michael Beneter. If you look at this image here, most importantly, the green dots are a healthy control, someone who doesn't have the disease. You look over time, and we're looking at the graph on the left, and we start to see an increase of this marker over time. Interestingly, it seems to match the increases we measure in more a qualitative way with what we had mentioned, the ALSF functional rating score. So it seems to be tracking in a similar way to ALSFRS. This is important because you have to try and understand it in context with proven biomarkers that are already being used in clinical trials. Even more interestingly, in a large cohort of patients that first of all carry the SOD1 mutation, Before they have the mutation, you don't see levels of this. They have the mutation. Before they get the disease, you don't see it in the urine. Once they get the disease, you see the increases. So it's certainly not an indicator early, but it is an indicator as cells are dying. So very interesting as a biomarker. I'm going to move to a few final concepts, and this we raised already, the concept of personalized or precision medicine, an initiative that is really big at NIH and really supported by the government. Again, the issue that we aren't all the same, and nor are people living with the disease the same. How can we select out our particular populations that we are going to treat? And this is a wonderful partnership at MGH with GE, with ALS Finding a Cure, and with many other partners, including Clive Svensson and the New York Genome Center. What they are going to do is take 50 normal patients, 50 people living with disease, over a timeline, so over a longitudinal study. They are going to look at MRI imaging. They are going to take PET, which is a glowing marker that you can see. They've got a great image for inflammation in the brain. They are going to take the DNA. They are going to take the stem cells. And they're going to look at all these different systems and composite on them omics, genomics, all the different types of omics to try and understand what is the picture between these different people and how does it change over time. So a huge initiative. GE is a great master at imaging markers, and they're a great partner to us on this program. Our final topic is clinical studies. We have a lot of effort in this. We are not going to have time to talk about our clinical management programs, and an assistive technology program we're beginning to expand on and get together to put out application proposals for this. I do quickly want to talk about clinical trials, and we'll close on those discussions. We have, over the past almost 8 years, funded the Northeast ALS Consortium Clinical Trial Network. They are no longer Northeast-based. It's really a network throughout the US, and that shows you that. And by us funding, so remember that when I first came on board in 2001, we did not fund clinical trials. Our challenge is that clinical trials are very expensive. We had small budgets, and we couldn't afford to do that. Although we do not fund phase 3 clinical trials because we feel that's the responsibility of the industry partner, and they are in their 50 to 60 to 80 million to do a trial, so we would finish our ice bucket funds very quickly. We do fund phase 2 trials and have done over the last several years. But in particular, we want to fund phase 2 studies that have a good biomarker profile, and I think I set the stage as to why that is so critical. We don't want a phase 2 trial to give us a hint of something, and we're not sure if that hint is real. We want to be sure that it's affecting the system we promised it should affect. It's tough. It's not easy to do. We asked for proposals. We got 12 in. We have some of them that look promising, but not one of them that the review committee would like to fund as they stand now. So the ones that are top have been notified that they have to revise their strategy. We will work with them in a very short time frame to help move it forward. But it's got to be something we can trust, that at the phase 2 trial end, it actually means something. And it doesn't get us on a path where we think we have a promise and it's nothing. So this is a very interesting study we've just recently announced, and I don't think I can describe it nearly as well as the clinicians involved. But I want to make a couple of points with this. It's retegabine. It's a compound that GSK has approved for epilepsy. It highlights, again, the opportunity to look at other treatments that are used for other diseases in ALS, but it needs to have a readout. And so the background to this is there is a sense in some studies that shows that cells in people's that have ALS in their brain are hyperexcitable. There is a very fancy technology called TMS, transcranial magnetic stimulation, that can measure the excitability of nerves. And this compound perhaps can affect that excitability. And how was the study done? Because usually you want some proof of it in some system. Actually, it was tested in stem cells alone. It's not gone into an animal model. A bit risky, may work, may not work, but the rationale was very good. So these are 2 lines that carry an SOD mutation. And actually, when they did this readout in these cells, they found that these neurons from these skin cells were hyperexcitable. When they then corrected the mutation in that cell, they found that hyperexcitability went away. So they felt, at least in this system and in this experiment, that the hyperexcitability was due to that mutation. That's a fairly standard and good study to do. They then found that if they used this potassium channel blocker, retegabine, which is an anti-epileptic drug, that they could rectify or restore the normal firing of those cells. That's the data we have. It's pretty good, it's pretty interesting, and we're now going to see if it works in people. How can we do it in people? We can only start with a small group because, of course, the anti-epileptic drug has never been used in ALS patients, so we're going to test for tolerability. But we're also going to measure and develop a new biomarker that's not so often used in ALS, and that's the TMSO, to see if we can check the firing of the before and after treatment with retegabine. And at the same time, by the way, we will take skin biopsies from the people in the trial and develop the systems in a dish and go back again and say, can we measure that change that equals a change in people with the treatment? And so what we're trying to test in this very risky, very potentially high-risk, high-reward study is to begin to ask, do stem cells in a dish give us information that is valuable enough for us to move into a clinical trial? So we're very excited about this. This is a partnership with multiple funding partners. GSK is providing the drug. Harvard Stem Cell Institute, ALS Finding a Cure, and ourselves are providing some funds for the study. So it just shows that we can, with very creative ways and very careful studies, expedite important trials which hopefully will have very meaningful data. It might go one way or the other, but then we know whether we should pursue this further and be recommending this as a treatment for ALS. I list here, I don't have time to talk about them now, I list here a series of trials ongoing, nearly ending. Clinical trials are a very difficult arena to navigate, and I know many people will ask, well, what trial should I be on? Why can't I be on a trial? We have today with us a clinical trials coordinator at the research, so it's really my moment to say, please join us at the research booth. I have a few colleagues who will talk about the work they are doing. Brian Traynor will be there, Amelie Gubitz from NIH will be there, and our clinical trials coordinator will be there. She is a source via email and phone call to ask your questions. She might not have all the answers for you, but she will work with our teams to try and direct you to the right place, to explain to you why a trial is not suitable, and to really help navigate that space for you. So please use those resources, and everything is on our website. I don't want to forget the numerous young investigators that have been funded through the Milton Safinovitz Postdoctoral Fellowship Program. We value, in fact, we have Marka Blitzvek with us today. I know she's somewhere in the room. She'll be at the booth as well. These investigators are the future for ALS. Both researchers, we fund clinical researchers through a program at the American Academy of Neurology. We have to invest in our scientists, and we have to treasure the commitment that they do every day. If you meet Marka and see her passion, you know that she's committed to finding a cure for ALS. These are not everyone. I always get nervous putting up things because you forget things. These are some of our partners from industry, from government, other ALS organizations. I value them enormously, and they are part of the team to find the cure. I can't help but just acknowledge many, many people that just in the last 3 months have dedicated their time to review, as I say, over 600 ideas. We are funding over 40 new things in the next couple of months, which will be announced. They are advisors to us. Ideas have helped us develop the strategic plan. Industry, clinicians, statisticians, globally, all over the world. I think there were 2 slides, and I may have missed the other one. Many, many people in just the last 3 months have been involved. But most importantly, and I didn't acknowledge any one single chapter throughout deliberately because it's so hard to be sure that I've captured every single one. All your chapters are incredibly valuable, whether your commitment to research has been small because of the limited funds in your portfolio or large. It's you that help drive the research forward, and it's you in the local community that have the connections and help bring us the partners. And it is the donors in the room, it is the people living with the disease, it's our other partners, it's the team together that can make this happen. And I'm so thrilled to be a part of it, and we will all be part of that cure, and we hope it's as fast as possible. Thank you all. And my apologies, Mark, if you had a sign, I completely ignored it. So I hope we're good on time. How much time do we have for questions, Mark? 15 minutes, fantastic. I did it right without knowing. So any questions, I will try and answer them. I might have colleagues who can help answer them, or there might be sessions later today that will answer them. Bear with me, I can't see very much in front of me because I've got spotlights, but there are 4 people helping you maneuver with microphones. I see a hand right there. If we can get a mic there. We have a mic there. Who's got a mic in hand? Go for it.
First off, nice job. Great presentation. Can you make this presentation available to the audience?
Absolutely. It's being live streamed as we speak, so everyone is seeing it and we can absolutely send it as a PDF, and I will leave it over to public policy to do that.
Thank you.
There was one coming up here.
In your opinion, can you give us the 5 most exciting things that you just spoke about? And the reason for that is that many of us have our database of donors. We would love to be able to go back right now to all the people that have donated to our walks and our snowmobile rides and our fishing tournaments and say, here's where in the Ice Bucket Challenge, and say, here's where your money is gone. And here's the exciting future. But we need that condensed so that we don't lose anybody.
So I suppose I put my 5 big things into 5 boxes for a reason. I think those 5 areas, so studies in drug discovery, we have 10 to 12 new ones which we plan on investing in. And we can send summaries and things once they're all finalized. Clinical phase 2 trial, very excited about the program. We will definitely have a few of those. They're 1.5 million each. It's already only a small amount we can invest, but we will have 2 to 3 of those, hopefully. The IPS core and the neurocollaborative, because they are open source opportunities for everyone to have access to. And then genetics. I strongly believe if we understand all the genetics of ALS, we're a long way ahead. So fantastic work at New York Genome Center and Project Mine, and others that I can't really talk about but will be coming up. So I can help with our development team to prepare materials that are helpful to you.
You talked about the influx of, that came in from the Ice Bucket Challenge. Who's deciding how and where those dollars are being appropriated? And then is your team part of that process in terms of saying, hey, let's take this chunk and put it toward, one of those 5 or others? Okay,
so the chunk that is dedicated to research is really something we spend with all, so I listed all those people at the end because they've been pivotally involved. Those are my colleagues with all over the world with different expertise. We already had some structured programs, so the 4 big initiatives that were announced earlier were all strategic plans that had been in planning but lacked funds, so it was a natural opportunity, and they had this exquisite opportunity of really bringing in, as I said to you, understanding the clinical side, bringing in people with the disease, making them central to discovering ALS through genetics, through the omics studies, through stem cells. So that's why those were chosen. I've had advisors, and of course internally with our organization, we've had strategic discussions. And so for each of the programs, we have a drug development program, as I highlighted, which is a solicitation of ideas from all over. The clinical phase 2 is strategically important. So we have a strategy set out for the next 2 to 5 years, which actually is going to be in publication and rolled out very soon, which will give a little bit of breadth of that. In amongst that strategy, we have programs that have very specific calls for proposals. I was very passionate that as I said to you from the beginning, there are many ways you can spend your money. I don't think there's any right way or wrong way as long as it gets data out in the end. However, I was very passionate that we had, and I've been with the association for almost 15 years and built programs. I didn't want it only dictated by what me and my colleagues around me and a few others might think. I wanted to be sure that everyone had a chance to submit their ideas. At that point, because we have spent the Ice Bucket 5 times over with all the things that have come our way, we had to have some level of system where there are reviews and oversight, et cetera. And that was already very much in place because that's how we've built our programs over the years. Hopefully that helps a little bit. Yes, I see a hand.
Hi. I appreciate that the association in terms of clinical trials is really trying to put their resources towards the phase 2 trials where a lot of the excitement and real challenges are. You talked about wanting to make sure we design the trials so we learn about what needs to go forward in terms of efficacy, but at the same time, when you talk about rigidibine, it's a small trial that's really powered for tolerability and for safety. And so I guess my question is, when we look at the literature, the biostat literature, there are many recommendations for how we get these two priorities to coalesce. But when we look at the types of trials Niels is recommending, it seems like we're heavily weighted towards the tolerability side and we're left guessing in terms of which ones to move forward. If you could just talk for a minute about how we fix that system that people have been talking about for the past 5 years.
So your point that the trials are listed as phase 2 tolerability, because that, truthfully, for that size is what you can really do with a small size. You can ask about tolerability or dosing or things like that. It doesn't mean to say at the same time we're not building in measures to see if it affects something, and that's why the value of adding on the biomarker and these are shortish time frames, and some of the studies where a biomarker is very defined, it doesn't have to be a very long study. It doesn't have to be the phase 3 long survival trial because it's the only approved biomarker that might be relevant. These biomarker trials are quite short, and they then will become much more informative to be able to design the bigger study in a much quicker time frame to answer the question you have. So yes, they have to say tolerability because it would be incorrect to say that those small size studies are looking for something efficacious. But by golly, if we get a good biomarker out of it, you can design a study much more effectively with probably smaller numbers of people, bigger than the phase 2, but smaller phase 3 trials that will very much dictate efficacy as well as things. So I think it might be perceived that they're all tolerability only, because that's realistically what you honestly can get out of it. But it is going to be a way of getting data to more quickly go to your efficacy trials. And also remember that we are an evolving clinical team over time. We've learned, and we've sometimes, as I said to you, because we are doing that now, we're forcing people to use biomarkers, it's not so easy. And the temptation, as we all know, is to try anything anyway, just in case it works. You know, one might say DEX at 80 million was a little bit of that experiment. We didn't have a way to measure if DEX engaged the target we thought was maybe mitochondria. We had actually no way of doing that. The company invested, and I'm not here to quote their reasons. We are grateful because that company is still with us, fantastic. But it was really an experiment that may have been doomed to fail. And was that hint in phase 2 really good enough? That's sort of the basis of how we have to think of changing things. I hope that helps a little bit. Yes. Oh, you need a microphone, sorry. And we have a microphone there, one moment.
Hi. I'm aware that some of these very exciting research things that you're talking about have been also undertaken by some other research groups, such as MGH has the biorepository, and ALS TDI is doing the iPS stem cells and also mapping genomes. How much sharing is going on and how much duplication of effort?
So first of all, the MGH biorepository is funded through our programs, and so it's very much under this umbrella. I didn't always exactly quote everyone, and it's just so complex. So the biorepository, which in fact we're about to announce an extension of that in Arizona, there'll be a second site for those. It's completely open source. Those are samples that are going to be accessible by researchers worldwide. One of the big things we've been pushing, and again, I failed to add in, we are working with all the teams to have a GUID, a global unique identifier, so that if a patient is in study X and study Y, they will carry with them that study tagged by that GUID. And so that can be then tracked, and you will be incorporated. If you've been in one study, we won't replicate that statistic, and we'll combine the studies. And we are actually also funding Neurobank, which is a huge databasing system that is tying all these different studies together so that we can logically, although there are many separate studies, analyze the data as one bigger study. As far as the iPS stem cell work is going, currently we don't have a direct collaboration with TDI. We're very open to discussing that. I think there is no harm in having multiple hubs of these things because it's a huge volume of work that has to be done. It's a huge cost involved. I think the bottom line is coordinating these studies. And I do believe that the GUID and all the programs that we are funding and setting up are open source in these particular areas of reagents we're getting from people. They are to be shared, they have to be shared, that's part of the contract, and they will be under a common identifier so that they will all be linked together. I hope that helps. Yes?
Can you hear me?
Yes, I can hear you.
Okay.
I'll repeat the question if it's a little quiet.
Okay. Can any patient be tested by the New York Genome Center?
So, no, can any patient be tested by the New York Genome Center? No, it's not a testing facility as such. It's part of studies. So if you want to be part of a study, and we will be improving how we communicate all the studies going out because there's just so much going on at the moment, we will make sure. But the studies are linked in with teams of investigators. So the New York Genome Center is responding to that study to do the sequencing and analysis. It's not a place where you would just knock on the door and say, I want to be tested. That said, once we have outlined clearly where all the studies are and the sites that are collecting and the centers, probably getting in through your clinical neurologist will be the way to go in.
Okay, I was curious. I have familial, and I have tested negative for C9 and SOD1. And to have my blood further tested was going to be over $10,000. So I have gotten in touch with the University of Miami and Northwestern. And Northwestern would really like to have my brain and my spinal cord when I do die. And I'm passionate about letting them have that. However, my question is, I can't find anyone who's willing to take it out of my body and send it to them. And I wondered if there was anyone you could put me in touch with.
Okay,
this is a big thing, apparently.
Yeah. So I'm surprised that Northwestern can't provide you that information because Dr. Sadiq is perhaps who you've been talking to, and Nyla. I mean, I think that they would be able to give you those clues. But if not, I know them well, and let's try and have a dialogue and see what we can do about that.
Thank you, yes. We've talked to the pathologist at Wake Forest, and as long as I don't croak on the weekend, we're good to go.
Yeah. Okay, so there shouldn't be I think that they should be able to work that out. So I'm not quite sure what it is that doesn't work at the moment, but I'll discuss it with you.
Thank you.
You raise a very good point, though, just before I go on to the next questions. With many of these studies, obviously, as part of the study and trying to identify the genetics, it's not as if when you get into the study, you get a little list afterwards of the genes that are involved, because it might be much more complicated than that. We have to first prove, we will never be able to go back to you and say it's this gene X, Y, and Z before we're absolutely sure, because as I said to you, we have many changes individually anyway in our bodies. And so if we are going to say that it's exquisitely linked to your disease, we don't want to make an error and say, well, this is the gene. So you are part of a study, and this is obviously everything that will be discussed with you when you get into the study, but it's not as if it's a gene testing facility as part of that study. Just important to know. Yes?
In an ideal world of science or ALS science, you'll be looking at a target a drug which targets both the upper and the lower motor neuron, the dendritic spine, the axons. Do you have any potential targets, any list of, potential candidates for this?
So that's a good, so your question is, what are the targets for the upper motor neuron system in particular?
Both the upper and the lower.
Okay, so I think those are good questions, and I don't think there is a specific list of targets that are clear. I mean, obviously, there are the common death pathways that we think about that might affect upper and lower, but it's a good thing you mention it, because a lot of our work has often been on lower because it's easier to do those studies. but there are some great scientists, one in particular, Hande Oslander and Jeff Macklis, who are working at University of Chicago and MGH, focusing on the upper motor neuron and trying to see if they can fall out with targets. If you're particularly interested in that area, I can give you contacts to connect with them. But I don't have an easy list, unfortunately.
So do you believe, the next question is like, do you believe in dying forward hypothesis, or is it more a dying backward hypothesis?
Yeah, that's a good discussion, and I have to explain it. That could be a whole scientific debate. So the question really is, and I brought up the fact that the muscle and the nerve cells are key in the disease. I don't know if it's more an academic debate in my mind whether it's that way or not. So the question is, does disease start in the motor neuron, or does the disease start in the nerve cell? It's very hard to know in people living with the disease because the disease has already started at a much earlier time point. We have some interesting data from mouse models to say that maybe it originates in the muscle and then it's the nerve. But my argument is that maybe it's a combination because it might start there, but it might have been a cue from the nerve cell that caused the muscle to have that problem. It's a chicken and egg question, and I'm not absolutely sure that that fundamentally will change what we do. I think both ends are important, and figuring out how to treat both is critical. That's my view, not everyone's view.
Lucy, we're going to take 2 more questions.
Okay.
To remind everyone, we will be having the Meet the Research session coming up following the next presentation, so you'll have more time to ask questions to Lucy and other researchers. Following this, we will have a 30-minute refreshment break, but we will be starting the government research session promptly at 11 for everyone that's watching from home on the live stream. So we have 2 additional questions.
Hi, you had talked about that a treatment will most likely lie in the cocktail or combination drug approach. Do you think that really tech will be included in that combination?
So probably. I mean, I think most trials are, so it in Europe, almost every person is on riluzole. It's just the way it is. So any European study, they already have riluzole. And for some, it has a modest benefit. Others feel not. So the trials are always anyway arranged with on riluzole, not riluzole. So yes, until it's proven that it's not helpful, I think it is a reasonable thing to include. Again, it depends on tolerability of the person, if they feel that they really don't feel great on riluzole and it doesn't. But in terms of trial design, it will always be riluzole plus, at least certainly in Europe, and often it is the case here. Last question.
Whatever happened to NP001?
So I can briefly say that we are looking at applications for doing a phase 2 trial on that. We haven't finalized review of it. NP001 showed a modest effect, not entirely statistically significant, depending on whether you'd used historical controls or not, so quite complicated. There was a hint of something. And what was particularly interesting is it seemed that they identified a population that would be more likely to respond than another population. So it fits very well as a phase 2 to develop the biomarkers. And so we are reviewing that to see if that's something to bring forward. Okay? I think that's it. Oh, I have waving hands, but I don't know if I have time for any more. Tell you what, we'll connect afterwards, if that's all right. I'm sorry, I want to be respectful of other presenters in time. Thank you all so much for your attention.